Hello, I'm Riley Grindle, and I'm a bioinformatician, a part of the Comparative Genomics and Data Science Core at MDI Biological Laboratory. MDIBL is located in the tippy-top corner of the US, just off the coast of Maine. And we're a small nonprofit research institute that recently transitioned to using AWS cloud resources rather than local on-prem compute. And in doing so, we stumbled across Nextflow and NF Core, which we decided to employ in order to handle an increase in the volume of next-generation sequencing data that was coming through our pipeline. And so MDIBL primarily focuses on aging and regeneration research. And one of the biggest problems that we face is the handling of the axolotl genome. I don't know if any of you are familiar, but we deem it an 800-pound gorilla, as it's about 11 times the size of the human genome in base pair comparison. And so to handle this behemoth of a genome, we've designed a pipeline called TXM Update, which you can see in the top blue section there, which is a transcriptome annotation refiner. So it utilizes upstream components of the RNA-seq pipeline to handle QC and alignment. I'm sure many of you are familiar with that pattern there in the first two quadrants of the pipeline. But then it diverges to perform a series of band manipulations, transcript scaffold assembly, characterization, GTF reinsertion, and post-processing. And that allows us to define much more accurate feature definitions for our axolotl, which is going to improve all of our sequencing experiments at the lab. And now that we've got more accurate definition, we also handle a lot of single-cell data at MDIBL. And so we've designed a pipeline to wrap around what I'm sure most of you are familiar with, some of the core processing components of a single-cell experiment, which are QC, normalization, principal component analysis, clustering, dimensional reduction, et cetera. And we call that pipeline SCScape, which really just allows us as bioinformaticians to streamline the initial data processing and ask much more complex questions much more efficiently. But yeah, I just want to give a thanks to Sakara, the NF Core, Nextflow platform for continuing their dedication to the community. So thank you all.